So why don't we so I'm just going to throw this open. I mean, I can find things to talk about, but I'd much rather find things to talk about that you want to discuss. So folks, who wants to start off with either a question for the panel or a topic for discussion? Do you have something that's on your mind particularly? Have you had, I'm particularly interested in hearing about your experiences locally, trying to do sequencing, trying to do genome sequencing analysis. It would be cool to hear about any successes, any challenges, any difficulties. Okay, so who's going to start? I might pick on you all. I'm going to pick on you all to turn your cameras on again, because I'm not even sure if everyone's here, because most people are keeping their cameras off most of the time, which I understand. But I'm going to ask you all. I'm going to put it in tiling mode so I can see. I'm going to ask you to pop your cameras on and just check you're actually here. Oh yeah, some of you are. Okay, good. Nice to see some faces. Many of the comments, many of the questions have been from a small number of people. I would love to hear from other people. Who wants to ask a question in real life of the panel? Oh, we're shy lot today. Who out here would like to discuss some issues they've had with data analysis or with COVID sequencing? Put your hand up, or just jump in and speak, or pop something in the channel. All right, we've got a question in the chat from Artem. Artem, it's always Artem and a few other people, but we'll have your question, and then I have some new questions or new topics from other people as well. This is actually going back to yesterday's topics in the morning, which was about different PCR primer sets. So we've got various primer sets, such as the Arctic method that we discussed, and there's also the Midnight set. There's one from NEB, I think, called Basket. Who would like to proffer any opinions about different primer sets and whether you should use them? Obviously, this is the Arctic workshop, so we're probably going to tell you that the Arctic primer set's the best one to use. But certainly the Midnight one. The Midnight one is called Midnight because it's a 1200 base pair scheme. Get it? Midnight 1200. So it gives you slightly longer amplicons. Anyone use the Midnight scheme? It's also the one that the ONT package. Anyone tried that out and wants to venture an opinion?
Hello, Nick. Hi, it's Gustavo.
Hi.
Hi, how are you? I just have the Midnight kit, and I have not used it yet, but I want to use it for wastewater. And that's one of my, questions, if it's good or not. And the other thing, how will we deal with Omicron? I've been doing basically a year of surveillance using qPCR, and most of my sites, they are between Delta and Omicron now. We are at the north side of Canada, and we're always a little bit late in all the variants that are coming from the south. I think beginning of December, we start with Omicron, but we're still having Delta in some places, like in other places of Canada has been completely replaced, and Delta has been extinct. So I'm wondering if I need to make some modification on the kit in order to have no dropouts, as I've been reading from different papers that when you use the Midnight, the primers that they are there, probably they are not the best ones for Omicron at this moment.
Okay, all right. Well, I mean, the best person to answer these questions is Josh, but I don't think he's here. So I can answer.
Can I jump in? I do sequencing of wastewater.
Oh yeah.
Done a lot of testing of different primer sets, and what we find is the actual size of the amplicon is kind of somewhat related to your success rate. So 1,200 base pairs is probably not good for wastewater. We found generally the Nimogen kit, which is about 200 to 250 base pair amplicons, works better than, say, the Arctic 400 base pair primers. So I think basically its size of amplicon is probably your
Yeah.
better thing for success.
Yeah. What's the name of the kit?
Nimogen.
Nimogen.
And they've recently redesigned their primers to improve their coverage of Omicron variants.
Okay, thank you.
Yeah, I was
I think
something similar to Aaron. Oh, sorry.
You go on.
We've used a similar kit from another company called Paragon, who also do 180 to 200 base pair amplicons, and definitely on wastewater, we have in some cases struggled with. We haven't used the Midnight primer specifically, but trying to do like our own custom, much longer primers for longer regions. We've struggled a little bit with wastewater for those longer amplicons, and also sometimes with clinical samples, like even clinical samples, nasal swab samples, the very small differences even between the Arctic and the basket. When we, between those two, we're sort of two partner labs using those two different primer sets, and we see even slight differences in our ability to recover complete genomes with those. So definitely sample quality and amplicon sequencing play together really tightly in between, whether it's saliva or nasal swabs. There's so yeah, I think it's kind of fit for the sample type is really important here.
Yeah, that's really useful feedback. I mean, we chose I think Josh talked about it yesterday. We chose a 400 base pair scheme because that was something that would work pretty well with high CT values. So CTs of up to 30, maybe a little bit higher, and it was compatible with ONT sequencing where the fragments can't be too short, and Illumina sequencing where the fragments can't be too long. So that was the choice of 400. And certainly fragmentation and degradation are related to CT values often. So, CT goes up as, RNA degrades, and it fragments as well at the same time. So having a shorter amplicon scheme is typically better for higher CT samples. So that makes perfect sense in terms of the wastewater. The Arctic 4.1 primer set is an Arctic 4 and 4.1 are kind of optimized to reduce primer dimer formation, which is something that Josh mentioned. That increases efficiency, so it should be more efficient than, for example, Arctic V3 on the same CT values. But yeah, the Nimogen is one of the shorter ones. AmpliSeq from Ion Torrents and one of the shorter ones. We have looked at developing really, really short ones. I think it was Mike Warraby came up with a protocol called Jackhammer that he used for sequencing the kind of patient zero HIV samples that were out of the freezer that had been stored for, I don't know, 100 years or something, and where the fragments were expected to be incredibly degraded. In that case, you use like really short amplicons. Well, short amplicons are not great for like routine use because they're very hard to balance and it's very hard to get them all to work. But for really, really fragmented stuff, it works well. So the flip side of that is Midnight, which is 1200 base pair scheme, probably doesn't work as well with higher CT samples. But the flip side is because it's got a small number of primer pairs, because it's got bigger fragments, when they work really well. So they balance out better, and you get more even coverage and more complete genome coverage at lower CTs. But then it tends to perform less well at higher CTs. There was actually a paper on this recently from some participant in the audience who's not here at the moment, called Bede in Oxford, looking at exactly that. And then your question about Omicron and Delta. V4 was really a reformulation to cover up all of the important circulating variants, including Delta. And then V4.1 was a refinement to sort out the dropouts that Omicron caused. So we're having to tailor these based on the dominant variants. And I think Jeremy's posted in the chat, there was a similar dropout in the Midnight panel associated with Omicron, which I think there's a fix for. Is that right, Jeremy?
Yeah, I've just quickly looked at, because I remember Nikki Fried had mentioned this a while back. But it's just to say that for the Midnight panel, in fact, I'll just copy the link directly to her Twitter post, which explains it quite well. And there should be a solution in there if that helps. admittedly, I haven't worked with the Midnight panel, so this is more so based on what she said rather than me.
Yeah, I mean, this is both the strength and the weakness of these PCR amplicon schemes is they're very, very sensitive, and they're quite cheap because you only need, you get all the reads are on target compared to metagenomics, and you get a large number of reads from successful amplification, and so you can multiplex a large number of samples on a run, and you can get the cost down to a very low number. But the trade-off there is they do need to be reformulated from time to time as mutations knock out primer sites, and there's not an easy way around that. As recently, PacBio announced a sequencing kit called Hi5 Viral, where they use a slightly different technique, and they actually use huge numbers of primer pairs, overlapping primer pairs. It's quite similar to the Arctic protocol, but instead of using one primer, they use tens or hundreds of primers in that same region, and hope that they get a good, well, they should get a good match by chance, and they think that might be a good system, but we haven't quite managed to evaluate that ourselves. So maybe there will be future ways around having to reformulate, but there isn't. Does anyone else want to comment on that, or there is another question in the chat? Okay, so there's a question here from I'm not AB Hidji. I'm not sure that I'm not sure if that's a name.
Hi, yeah, it's Abhishek.
Abhishek.
I didn't. Yeah, just abbreviated everything in the Zoom, and I don't know how to change that here. So hi, yeah, I'm working a bit with GISAID metadata, and it's obviously the metadata is it's quite varied. There's the dates are okayish, but some dates are not correct, and you have, especially if you look at say gender and age, it's a complete mess. So just wondering if there is any people working with metadata. Do you have existing pipelines? So is it something that would be good to have as a public resource? So yeah.
I think many of us on the panel have strong feelings about this question, and it's certainly Rachel's laughing because I think we've spent a lot of time having to deal with these kind of issues. Rachel, do you want to take that one first?
So I think in response, okay, this isn't working.
Go ahead.
I think in response to that, I'm not doing. I have a script within DataPipe, which is the Cog UK pipeline, which it does basic cleaning enough to make the pipeline not fail, but it doesn't actually do anything in the way of trying to improve on the metadata, problem solve. I think if someone were to come up with some public resource that spotted people who'd put in dates the wrong way around, month, day, rather than day, that would be amazing. But all we do is say, if it doesn't fit our spec and isn't a valid date, throw away the sequence. There's quite a lot there on GISAID. So actually we can afford to be a bit brutal with getting rid of sequences we think the metadata is wrong. And the other thing I found is that on GISAID, they do allow you to put accents and things into place names. And some, if you use those place names for labels in any way, that can break. So yeah.
I think it's more of an issue for smaller countries where you have much fewer sequences, and not like US or Germany, which you can drop 50% and you still have a lot of good data. But okay, thanks.
I mean, there's a general point here. I mean, certainly we find that sometimes the errors are obvious. Like, so we see a lot of errors. We see a lot of errors in the UK metadata coming through, and often they're easy to track down. So you said that transposition as Rachel's talking about, between month and year or something like that, and that's easy. But actually, when we dig into some of them, they go all the way back up the chain to the original kind of canonical information for that sample. So they go back to the original clinical database or sheets that someone's written down, and it's actually sort of impossible to then do anything more with that because that you don't have the information. You know it's wrong, but you don't know what the correct version is. I think there is a problem. Maybe it's worth a little chat about it. There is a problem, which is that we fix those, or we try and fix those where we can. But quite often in the UK, we flow our data into GISAID and ENA really quite quickly, you know, days or up to a week of lag, and often that's detected after that time. And then it's kind of very hard to go and fix, and kind of, you know, no one can really be bothered to go and fix it, you know, in the databases, because there's not an easy way. So we can fix it in our database, and it's on our products, our analysis products on the website, etc. It gets fixed, but it's hard to go back. We do in really egregious kind of situations, but we can't fix it in the main. And I think it would be nice if people, you know, I think it would be nice to have a system where the databases were, I guess, easier to update. And also, it's probably important for this course, it's probably important to just get across that idea that it's low, but it's a significant rate of errors in terms of metadata. So it really needs to be borne in mind when doing analysis, you know, because people say things like, oh, you know, Omicron's been around for it's been around for a year, or it's been around for 2 years because I found a really old case. But of course, much more likely to be a metadata error than we missed Omicron, you know, it was there 2 years ago. So I don't know the panelists, have you got any ideas about how we can do better on this? It's a hard problem to solve, I think, especially with 7 million genomes. No one knows how to solve this problem.
I think.
Anyone?
Please go, JC.
I think there are a couple of resources that are out there, but I don't know that. So there are like sites that should be masked. That I think there was a virological post early on that started a list, and I think that's been kept up to date. And I think like the NextStrain repository keeps a list of sequences that are excluded for quality reasons, and so I think everyone's sort of doing that on their own, but I don't think that there's a central repository of it. And I think I don't know, it's very important, but it takes a lot of work, and it feels sort of like thankless work to keep a list of sequences that aren't good. But
at the same time, you want to filter based on the things, metrics that matter.
Yeah.
So you filter for a specific variant that you're investigating, and you'll filter by date, and you'll say, for this tree, I'm building Omicron, I think sequences before this date are unreliable, or sequences where the lag time is greater than this are unreliable.
That is something we've begun doing, is filtering based on the time between collection date and published date, because if it's unreasonably long or negative, we can probably get rid of it. But again, as you say, if it's for a location that doesn't have a lot of samples, then it is important to think about.
Thank you. Yeah. Okay. I mean, yeah, we're not going to solve the metadata problem today, unfortunately, but it is really worth understanding that these are imperfect systems with data collection, often done by humans, and often passing through multiple different processes and systems, and not always in a completely automated way. So there are lots of opportunities for mistakes to creep in. So just being aware of that, and I think hopefully if this course has given anything, understanding where some of the sources of variation can creep in terms of sequence data as well, is an important thing to have in mind when you're doing analysis of kind of other people's data, if you like. Okay. Do we have a bit of discussion around CTs in the chat? I mean, I guess a related question is where can, from Li Qing, where can you find where can find the largest collection of genome data associated with good metadata, great metadata? I'm sure great metadata is going to be possible, but so, you know, where's the best canonical source of genome sequence with well-curated metadata? I guess he's making the point everyone's spending a lot of time having to clean stuff up, and that's not a good use of time. Is there a good source of a kind of curated data set which has a higher confidence? I think we have something like that in COG, but but is there a public source of such a thing?
COG is probably, if you only care about things coming from one country, or don't care about things coming from one country, we probably do have some of the more stringent cleaning and careful curation of our metadata, and there are a lot of sequences, but it's not going to be representative of the global diversity.
Yeah.
What are people's, so I do a little bit of the analysis, but not as much. What are your sort of feelings on, my instinctive reaction was NCBI GenBank, because I know they do a lot more quality control. What are your feelings on using that versus GIS8? I mean, I know for the sort of scale and speed, GIS8 is obviously the one, but if you're looking for something with more maybe QC'd metadata, does anybody use GenBank routinely?
I've not used it, I'm afraid.
Yeah, I mean, GenBank has got reasonably good coverage of certain countries and then kind of nothing from others. So it doesn't make a fantastic global data set, but it's got certainly I think all the UK sequences and all the US sequences are in GenBank, but I'm thinking that it's not got brilliant global coverage. And I think it's only maybe only a couple of million sequences in GenBank, I'm thinking, but
it's definitely less, definitely less total, and but that's a good point about it being sort of patchy and less represented in specific places.
Yeah, I mean, my impression is not that easy to interrogate that database, and actually the metadata is almost not as rich from GenBank as it. GIS8's actually got quite a lot of metadata fields in a flat format, just because they've got a kind of simpler task of just dealing with viruses and a couple of viruses, so they have that, the strength is getting that big metadata table out and everything being flat formatted, and I think it's quite hard to do that. I think it's quite hard to do that from GenBank, although I haven't really dug into it recently. Okay, so there's quite a technical question for JT about Bayesian inference, which maybe you can answer in the chat. Lucien is also asking a question about swabbing, which I think is an interesting one, but probably off topic. Anyone else out there, done some sequencing and run into troubles, and would like some advice from the panel, or is it thinking about doing sequencing but is worried about how to get started? I'd really like to hear anything from the audience along those lines. If we don't get many more questions, we will just wrap it up. So which might be fine for everyone, but if you do have any questions, now is definitely the time to ask.
Well, one of my questions is, what is the cost? How you can scale up and reduce the cost of sequencing. And my second question is with bioinformatics is do the analysis. We have tools that they are provide generously for a bunch of people they are working, but one of the costs is access to the hardware, power computing, and what's your opinion to use between a cloud system and have your own system in home? I suppose there are some pros and cons between each one of those, but what you would recommend it as a long investment?
Okay, both good. So two good questions there. I think it's a relevant question because this is an Arctic Climb workshop, and obviously Climb is a cloud computing infrastructure that we've used very heavily in the UK, and it supports some of the services that we've talked about today. But Andrew, I think we wanted to pick up on your first part of your question, which was about the kind of cost of on scale of sequencing.
So if you're doing a lot of sequencing, I only have numbers for Illumina, but it's about £5 per sample. So that is from extracted RNA to plus Arctic plus sequencing. But that's only if you do, thousands at a time. Obviously, if you're doing smaller batches, then the cost goes up quite a bit. But we've found that you can actually dilute a lot of the reagents, and you know, if you don't follow the manufacturer's protocol, you can save a lot of money.
Yeah, so you obviously have the Corona hit method, which was optimized for scale and cost, wasn't it? So you can look at the Corona hit paper in, I think, Genome Biology for information about that. Is that still your current method?
Yeah, and as time went on, we made lots of optimizations. Actually, a lot of them were accidental optimizations, where people accidentally put the wrong stuff into tubes, or too little, or whatever, and then they found it just worked. I mean, at the very beginning, we were doing, say, everything on ice, but then we found when we had no ice one day that it didn't make any difference. And particularly for RNA, people, you know, put the samples back into the freezer and out again, in and out. But we have an experiment running at the moment where we've just left samples on the bench for a couple of months, and we sequence them periodically, and there's no change. So yeah, just experiment with things. I'll put
these protocol optimizations, are you tracking them online or something for people to look at, or not really?
I suppose they're internal. But we share them with a few other groups in the UK that are doing sequencing still.
Okay, so that's a potential resource. Would anyone like to talk about the second part of that, which is basically, I guess the question is, you know, whether you should invest in computing hardware locally to do sequence analysis, or use the cloud, or use some kind of combination of those two approaches. What are people's feelings, you know, if people are setting up their own sequencing facility for their region or their country? How should they deal with compute?
That's such a wide, difficult question. I mean, I feel like the party line, given this is a party line workshop, should be cloud is great, use cloud. But obviously, it's so situation dependent. I mean, the Nextflow pipelines, for example, will run on anything. You know, it doesn't require a GPU or anything like that. So you can run it on your laptop. It will just take a very long time. So yeah, it just so depends on your own personal circumstances. I could certainly see in different areas where you don't have such good access to sort of institutional computer processing, like you might at a university or something, sort of a local machine could make a lot of sense for doing jobs like this. But
I mean, it may be illustrative to say how we do sequencing and analysis in Birmingham. We've mainly used Oxford Nanopore sequencing, and if you use, we have a GridION, and the GridION is a pretty good platform for doing sequencing. It can run 5 flow cells at a time, and you can probably get between 48 and 96 samples on a flow cell. So it gives you reasonably high scale. You could do a couple of thousand a week comfortably on a GridION. And we found with the GridION, you can do the analysis locally on that instrument. So you can run the Arctic pipeline on that instrument and generate your consensus sequences. You can run Pangolin, get a lineage assignment, Scorpio, get a variant assignment, and have enough information to kind of answer the basic question of kind of what you have. I think at that point, the difficulty becomes, if you're interested in doing more complex analysis, or you want to put things in the context of the entire data set, we would then move stuff onto Climb, which is our local cloud computing service. And we actually have all the other genomes are there, sitting there ready in trees that Rachel's pipelines generated in order to contextualize that information. So when you're dealing with storing all of the data and analyzing all the data at scale, it then becomes quite a sizable computing task. And so if you can use someone else's infrastructure like Climb, or use cloud infrastructure, that's obviously a lot easier to do. But some places will have their own local high performance computing that you could also use for that kind of task. So cloud is just someone else's computer really in somewhere else, right? It's no different from local computing in terms of the setup. Something we are trying to do as part of the Climb project and part of the Arctic projects is make more of these tools and databases available. I mean, Rachel, can you remind us how the situation with Civit is? Because Civit is a great example of a tool that like leverages having Climb churn through all this data and spend a lot of computation, but then in theory you can take that product and use it with Civit to do the analysis that people commonly want to do, which is how do my new samples sit in a global phylogeny. So Rachel, what's the sort of plan for that, do you know?
So Civit 3, which is the currently available main version of Civit, it uses GoFaster instead of a phylogenetic tree as its way of picking out catchments for your sequences. And on Climb, within the COG UK setup, we pre-compute the file that is needed by GoFaster. So GoFaster has a command, GoFaster up down, that would generate a row of data for every sequence in the global and UK database. And so that's already done if you're working on Climb within the COG UK setup. But if you run Civit and that file doesn't exist, then it will be created by Civit. So that first time you run it, when that file is generated, it'll take a lot longer. But if you have multiple commands, then you can reuse that file when it's been made to save time so that you're not repeatedly doing the hard work, if that makes sense.
So is it possible to, at the moment, you take Civit and then take the product from on the COG website and use that for your analysis? Is that, can you do that? Or
so not from the COG website, the outputs that we publish publicly from COG UK do not support directly, they don't directly support Civit. You probably could use them, but you'd only be getting the information for the UK data because we're not allowed to make the GISAID metadata available. Within the Climb setup, we have done a lot of the hard work on the GISAID data and metadata as well. So when you do a query on Climb, you can already access that information, but you'd have to do that or have that information downloaded where you are as well if you're not able to access Climb.
So hopefully maybe in the future that will be something we can offer out as something that anyone can use. I don't think that would be so popular.
It might need agreeing to agree to it.
But it also does kind of just bring out an issue, which is quite an annoying issue actually, which is that GISAID, whilst it is a very big database, useful database, they do place quite significant restrictions on reuse of data, which does kind of make life a bit of a pain, because it would be nice for us to be able to put that database online for anyone to use, and it seemed like, you know, it doesn't seem like a big problem to me, but we would be breaking the terms of service of GISAID if we did that. And I think that's, you know, the benefit of GISAID, I suppose, is that it gives some protections that people's sequences are not going to be just, you know, used without permission, but it also has some drawbacks in that it makes certain things that kind of should be easy quite hard to do. So that's maybe, I mean, I know that Usher has got around this problem by having trees and things that are generated just from the NCBI information, and maybe that's a possible way around. Obviously it's not the whole data set, but it'd probably be good enough to give you a useful result maybe. Okay, any more thoughts on where you should do your compute? I mean, Arctic and Climb is a nice combination of projects, because Arctic is really about empowering sites to be able to set up their own sequencing. That's partly why we've focused so much on Oxford Nanopore, because it's obviously a relatively inexpensive sequencing platform in the form of the MinION at least. And of course, other instruments are available that are relatively inexpensive to get up and running with sequencing. And our experience doing training courses around the world is that, you know, certain cloud services are just not that accessible to people because, for example, of issues around available network bandwidth and the cost sometimes of actually using those services. So Arctic is very much about doing sequencing locally and doing some analysis locally. And Climb is very much about doing things in the cloud kind of centrally. And to my mind, those two approaches are actually, they work hand in hand. You know, you do the right thing, you do the right thing in the right place. And so certain types of analysis are great locally. Buy the instruments quickly as possible. Don't need to upload all your data. Don't worry about the kind of privacy of the data. It's all stored on your infrastructure locally and done quickly. And then move off the data sets for the global analysis to the cloud. Seems like a model for the future.
Yeah, I was just going to add, sorry, a little similar to what you just said, but yeah, as a non-Arctic Climb person or someone based elsewhere and a little outside the system, I was just, for what it's worth, going to add, we do a very similar thing. So, you know, local analyses when we can and when it's appropriate. But so I'm based in the US, but a lot of the work we do is sort of collaboratively with a number of partners in West Africa, where we've set up sequencing capacity and also sort of analysis systems there. And we have generally and kind of for several years now used cloud-based platforms. And I think it is sort of, as Nick pointed out, it's a balance between, you know, we do run into challenges of internet connectivity issues sometimes and access, which is definitely a downside of cloud-based platforms, depending on where you are. I think the upside that we found to cloud-based platforms are two things. One, you only, if thinking about costs, sort of it can be more expensive per unit, but if you're not sure what usage is going to be and you're just sort of exploring, you know, you only pay for what you use, and that is an advantage that it allows you to kind of scale up when you need to and retract when you're not using it, rather than that big upfront investment in sort of your own sort of local server or whatever. I think the other aspect that we found good about cloud-based analysis, particularly for COVID, when things are moving really fast, is it's been easy to share protocols and share pipelines and run the same things collaboratively when we're not able to be in the same place as each other. So just for what it's worth, those have been our kind of experiences with cloud-based and why we do it where we can, although there are occasions where internet doesn't allow it.
Yeah, no, that's great. That's great feedback. Yeah, I think Broad use a platform called Terra, don't they?
Yeah, we use Terra, which was developed by the Broad and Google, which we use for our analyses, and many of the partners that we work with are using also, and it's being increasingly used elsewhere. It's I guess maybe I don't know so well the Climb system, but Terra is you know, I believe anyone can make an account and you can use it, and it has quite a nice. I think one thing I found good about Terra is it is relatively easy to like bring in other people's workflows and run them yourself without needing too much kind of coding background and things. So if you can find the workflow that kind of someone else has already shared one that does something like what you want to do, you can bring those in and run them relatively quickly with your stuff. And I imagine others have very similar platforms like that.
Yeah, I mean, Climb is mainly a UK, so it's free for UK microbiologists working in academia and government, but we are looking to hopefully extend out some, you know, access to certain pipelines and certain infrastructure to the whole world, you know, for SARS-CoV-2 analysis. But because there's such a demand for computing resource out there, we have to be quite careful about what we offer, because we don't want to offer something and then have it inundated with use and we can't support it. We don't want to offer something we can't support in the long term. So we just have to be a little bit careful about, but a lot of things you use, I mentioned already, like for example, the Pangolin service actually runs on top of Climb. So we know we do offer these kind of free to use services. One thing I've always wanted to do is offer the Arctic bioinformatics pipeline through Climb to anyone that wants to use it, such that it is available. And hopefully this year we'll manage to do that. But we've last couple of years we've got rather sidetracked into sort of dealing with the UK situation. Okay, I'm just looking at the chat in case there are any more. Is there any more feedback or comments from anyone on anything that's come up? Okay, I'm looking at the comments. I don't see any more questions. I think I'm going to start to wrap this up. So I've got to just do some housekeeping bits now. So the first thing is, yes, all the videos will be available from all of the talks, and we will put them on the course website, and they will be publicly available as well. And we'll do that as quickly as we can. It usually takes a couple of days though, because we have to edit the videos and upload them. So please look out for those. So if you missed any talks, or you want to revisit the talks because you missed some content detail, you can go back and do that. The other question that everyone always asks is, can they get a certificate of attendance? Yes, you can. But in order to get your certificate of attendance, you have to fill out our feedback form. So we can get more information from you about what you enjoyed, what you didn't enjoy, how we should change this course, etc. So I have pasted the form in the chat now, and I think that link will also be on the course website. Lisa can correct me if I've got that wrong. And we'll post it in the Slack as well. Talking of the Slack stays open, so you're able to post follow-up questions there. Andrew just said he'll post his protocol optimizations on the Slack. So if you're interested in that, you can keep an eye out on there. And Slack is just available. It doesn't cost us anything to run. So if that's useful for you, please continue to use it. All that really leaves for me to say is to thank people again. So I want to thank all of the speakers today, and all the speakers yesterday, and panelists that came back for your great talks. I hope everyone appreciated it. If you want to send some applause, then Zoom has a button for that. Thank you everyone for doing that. And I also wanted to thank Lisa and Mavis from the Climb Big Data team at the Quadrum for organising the workshop and getting you all here at the right time, and the registration process, etc. Without you, it would not be possible to do. So thank you very much. And we also thank the Arctic and Climb funders, the Wellcome Trust and MRC UKRI, for giving us the money to pay us to let us do workshops like this. So I'm going to leave it there. Thank you, everybody. It's been a great pleasure. I hope you have a renewed enthusiasm for SARS-CoV-2 data analysis, and let us know how you get on. Bye for now.
Thank you very much.
Thank you.